Thank you so much. I'm going to go ahead and share my screen now and get started here. Where is my... Okay. Sorry about that. And I don't know why I'm having issues with this now, of course. Goodness, what are you doing? I'm so sorry. Give me just a moment. Can you all still hear me okay?
Yes, we can.
Hello?
We can hear you.
Okay. Thank you. Okay, so my research is this is actually the ultimate goal is it's the environmental enrichment of bumblebee gynes and the effects of exhibiting foraging behaviors on the early nest initiation stage in a research setting. That is a mouthful, but basically what I want you to take from that is that I want to know how queens are affected by enrichment, particularly environmental enrichment, and this would be in the form of foraging behaviors. So just a couple concepts we're going to go through because this is developing research. I'm going to start with an introduction and objectives, bumblebees in the wild versus the laboratory, commercial rearing versus captive rearing, the beef box, experimentation and current findings, and then finally discussion and the future of the research. So the introduction. Why do we study bumblebees? We study them because they're very complex social organisms. They're very commercially available. They're incredibly important pollinators, both in agriculture and also in the wild. And for research, they're a very valuable specimen because they're easy to get and they're easy to care for. This particular species of bumblebee that I work with is called Bombus impatiens. It's actually from the East Coast, so it's not native. They're a very robust commercial specimen, which means that they readily start nests, they readily raise brood, and they are very easy to care for. In our lab, we study queen rearing behaviors, nest initiation, and brood care, as well as mating behaviors and molecular research. So I want to talk a little bit about organismal enrichment, which is not a new concept to anyone. We have pets, we have zoos, we have research. All of those have very strict protocols for how we care for an organism in terms of enrichment. The way that we allow them to live their lives, especially in a research setting, is very strict with higher organisms like mice, rats, dogs, apes, and monkeys. There's a lot of consideration that goes into that, but less so with insects. And particularly with bumblebees, they don't have any enrichment at all. And we don't know how that affects them overall as research specimens. So that was one of my early questions to my PI. I asked her whether or not allowing them to forage or exhibit foraging behaviors, whether or not that would change their lives in any way, or whether that would change our research in any way. And she said, I don't know, let's see. So this is where my research and my questions have led me. In terms of enrichment, we want to decrease stress. So whenever possible, we want to decrease the stress because that makes for a more productive bee. For quality of life, we want them to live as good a life as they possibly can, even as a research specimen, and particularly as an insect. The quality of research is really dependent on enrichment in my particular case because I'm interested in knowing how it helps or hinders them. Because foraging itself is a stressful behavior. So I want to see where that line is, where it's too much stress, but at the same time, I want them to live as close to their normal life as possible, even in a research setting. My questions for this were, do enriched bees initiate nests faster than the control? So does allowing them to forage make them better bees? Can this research inform our understanding of challenges of captive rearing? That main challenge I'll get to later. And can enriching commercially produced bees improve their performance as a research specimen? With these questions in mind, my research went forward with a couple other things. So bumblebees in the laboratory versus the wild, it's very different. So in the laboratory, we keep them in the dark. They do not see the light of day unless we want them to, or unless we remove them from their colonies. So what this means is that they always live in the dark, which part of their life is they are subterranean. Their colonies are subterranean under the ground. So living in the dark is not unusual, but living in the dark all the time is unusual. So I kept that in mind and thought, oh, that's a little strange. Another thing is, dependent on the experiment, their housing will change. So sometimes they're in these little cups that you see here. Sometimes they're in small nest boxes. It varies, but there's no enrichment there. They're provided food, their temperature control, and their the humidity is controlled. But other than that, that's where they live, that's where they exist, and that's how we conduct our research. Another unusual thing about the lab with our bees is that all of the casts live together. So all of the queens that emerge, they live together, all of the males and all the workers, and that is also unusual for bees to do. Usually in the wild, they'll be able to forage and leave the nest when appropriate. But time is also really of the essence with bee research, and I'll explain that a little bit later as well. Bees in the wild are dependent totally on species, but they geographically exist everywhere in the world. They are diurnal, which means that they forage in the daytime and they rest at night. They are very geographically diverse, like I said, and they're dependent on seasonality. So all bees emerge in the spring, and then they hibernate in the winter. This would be reproductive queens. All of the other casts, the males and the workers, they will die after the season is done. So at the end of autumn, kind of early winter, all of them will die off, and then the queens go into hibernation to emerge the next spring. Challenges of commercial and captive rearing. So I mentioned earlier that captive rearing always comes with its own challenges because we have to get the right time of the year. We have to get the right type of cast. So we have to actually go out and find these queens if we're actually going to capture them. And then we have to house them. So the conditions that I want to consider is that they're living in the wild, and then we put them in a laboratory setting and they just don't do well. They just don't. It's a very well-researched fact. Captive rearing just doesn't do well for bees. And I wanted to see why and examine the reasons for that. Commercial gynes we basically mail order them. We put in an order, they send us a colony, and then we do our research with that. However, we're not given the raising protocols, so we don't know what conditions they're raised in. We don't know if they have any diseases. And we don't know if there's a presence of inbreeding, which can definitely affect our research. The housing, like I said, is a box like you see in this diagram here. And it's literally just a cardboard box with a plastic box inside, and that's where they live entirely. And that's just, it's not normal for them.
Yes, us. Your bumblebees. Hello. Let us show you our eco-friendly house. The Natupol hive. It is eco-friendly and made of 85% recycled materials. It is a smart architectural design and is just one of the many eco-friendly innovations that comes from corporate's roadmap on sustainability.
From. I just wanted to show you the complexity of the box. It is a very smart model, but it still ends up not being natural for the bees. So I developed a protocol and a prototype called the Beef Box. The Beef Box is a bumblebee enrichment enclosure for foraging. So it's divided into three parts: the top enclosure, the lower enclosure, and the RFID connector tube. And the RFID, we adhere to our specimens on their thorax. And the aim is to know the frequency of visits up top and the return down into the actual enclosure. This is the design evolution and the 3D print builds that came after it. So you can see in the beginning, we have just the general model, no enrichment at all. In the second photo, I made a duplicate so that I could run two treatments at a time. And then finally, the final adjustments and enrichment were added in that last picture. And that is the model we're using today. Here are my experimental protocols and current findings. So some observations that I've made, here's my first bee that I ran, and you can see her RFID chip on her back. I wanted to know whether or not species-specific behaviors were exhibited. And yes, they were, because within 48 hours, that queen went into the top box to explore, to feed, and to forage. Caste-specific variations are kind of different. So queens will intensely look for resources and then initiate a hive, whereas the workers, once they emerge, they take over. So they'll take over for their queen, who will then stay in the nest predominantly, and then all the workers will bring the food back home. The male drones, once they're mature enough, should leave the nest and not come back because they're not meant to mate with their sisters or their mother. This is the experimental design. So we see this one is the control. That is the second one is the nest box with resources in the top and the bottom. And then the third, which only has resources in the top. The reason why I use this model for my initial test is because I wanted to know if the bees exhibited preference. So did they prefer to eat in the upper box or did they have to eat in the upper box? So this is what, this is my, my findings. So this gyne here, she's unmated, so she can only produce males if she were to initiate a nest, which she did not. Does she exhibit phototaxis? Yes, she does, which is normal for this species. Does she exhibit preference? Yes, she does. I ran another treatment of just 5 callow workers, and they did initiate a nest within 14 days, which is typical, but no brood emerged. They did not exhibit phototaxis, which means they did not even want to explore the upper box. And did they exhibit preference? Yes, they stayed in their nest where their brood was, which is normal for the species. So 2 key takeaways for my research thus far. There's been some unusual early behavioral patterns because I did expect the queens to explore, but I also expected them to initiate a nest, but she did not. In terms of my workers, they did not want to forage at all, which is a normal behavior for workers, but my workers did not have a queen. So we believe that that's the reason why they did not want to go forage for her, because they did not have one. But this is future treatments for further research. It's very complex because these living organisms are eusocial and there's so much variance in their behavior. So we're hoping to replicate until we can get some hard data for this. But in terms of future research, we want to replicate this experiment with mated queens. We want to have closer behavioral observational studies. We do want to try some captive rearing and see if our wild bees will do better in the lab when they can forage, nutritional assays, and neurological comparisons. We want to compare the brains of bees that are enriched versus bees that are not enriched. So that would definitely inform whether or not foraging is even valuable to a bee as a research specimen or not. So there's lots of considerations. It's very exciting. And I simply adore bumblebees, and everybody that knows me knows that. And I'm very passionate about this research and its many applications. These are my references. Do you have any questions? And thank you so much. Thank you, Dr. Woodard and Dr. Costa, for their continuous support. I also acknowledge and honor the Native peoples upon whose land I respectfully conduct research, both in the lab and in the field. Thank you so much.